Thank you very much and good afternoon. Thank you for the introduction and thank you to Clive for inviting me back to this great meeting. I haven't been back to Barbados for almost 9 years, so it's been quite an emotional return for me. I haven't left the hotel yet, so and there are about 100 people queuing up to meet with me in the next couple of days, but I thought I would focus on the conference for now, which is why I'm here. So I'm going to talk about cure, and I'm going to talk about concepts, but I also want to keep it real, because we can't be talking about cure when we're still grappling with access, But as an academic, we must be pushing the boundaries. And there will come a time when access will be available to everybody globally, and 90 or 95 will be a reality. And then we must start thinking about how to eliminate the reservoirs and how to cure people who are living with HIV rather than talking about AIDS, which, as Clive says, AIDS should not be a word that we should still be using in this day and age. So, very happy to be back in beautiful Barbados. I love this slide because it just demonstrates the color of the sea and the flowers and just the hospitality and, of course, the flying fish at the bottom there. But I left Barbados and I moved to Cape Town. This is the famous Camps Bay Beach, and you can see that it has lots of attractions. And that's not my car, but there's a Ferrari club in Cape Town, and they come and oppress us every Sunday at Camps Bay Beach. So I thought, this is the wine district of Western Cape where I live. I live and work, and South Africa is a paradox of exuberance and poverty. So. People talk about Cape Town and South Africa as being this highly developed part of Africa, but I assure you that there are some areas that even I can't go to because it's so impoverished and so dangerous. Oh, sorry. This is the teaching hospital I work in. Tygerberg Hospital is one of the largest hospitals in Africa. It's essentially for the underprivileged, where we treat 80% of the population of South Africa that are unable to afford health or insurance. So we're talking about care to cure, so I'm going to be looking for clues, and looking for clues in various aspects of the outbreak, in the origin, the animal world, the animal models of the old and the new world, the profile of the disease in man, some historical cases of cure and modern technology. But as I said, we really want to keep it real, but allow us to dream and be futuristic, because that's what academics do. We do need to make sure that we're not losing sight of the reality, and AIDS should not exist, I agree. It still does, and that's just an unacceptable situation. We should be using effective HART to minimize new infections, reduce comorbidities. Mother to child should just be, not acceptable. But persons living with HIV, the numbers will progressively increase. So we're going to have lots more people living with HIV. So what are we going to do with them? We haven't refined the art. We're just preoccupied with 1990, but there's a lot going on that, we just don't have the energy for. So let's go to the origin of HIV and see if we can find anything there that may help us. There is a retroviral party going on in Africa. You all know that. There are many simian immunodeficiency viruses, and they range across most of sub-Saharan Africa. That's the simian belt and the SIV belt. And here you have the chimpanzee. I want you to notice where the chimpanzees are. In Central Africa and on the tip of West Africa. And then the Sutimangabe, which is over there as well, overlapping with the chimpanzee range. So the SIVs, there's an SIV for every monkey. So there's the green, African green monkey. So we have SIV, African green monkey, sooty mangabe, mandrill, chimpanzee, CPZ. But there's no SIV for humans. Okay? It's very important to remember this. So over millennia, these SIVs have rotated through all the monkeys in Africa, and these are the old world monkeys. That's what they're called. The new world monkeys went to Asia, and they escaped Africa before the SIVs managed to circulate. And then somewhere along the line, we had some SIVs combining to infect the chimpanzee, and then the SIV from the chimp became HIV-1. The African primates over millennia have adapted to the SIV. So initially, I'm sure it wiped out the monkeys of Africa. But over time, they went through what's called a bottleneck, and they were able to adapt. And so the monkeys of the maybe 2 million years ago were suffering from an HIV of monkeys, and they were dying, but some of them had natural genetic traits that allowed them to survive. So most of them died, but a few who had the traits that would protect them from SIV, like HIV, would go through this bottleneck, and then they would populate the rest of Africa with a resistance to SIV. So this is the family tree of SIVs, and you can see here that most of the monkeys here have an SIV, no relationship to HIV, but the SIV that's related to the HIV, it's confined to the chimpanzee and very, very related. So SIV chimpanzee is very close to HIV-1, and therein lies the clue. So we know that the ancestor of HIV came from SIV. I won't talk about HIV-2 on this occasion. So the first confirmed case of HIV was in 1959, a European sailor in Central Africa. Now, a lot of you think that HIV started in 1980. It's not true. There were several small epidemics across Europe as early as the 30s. And there's historical evidence for this. And then when you try and date using genomics, you try and date the outbreak of HIV or when it first came to be, modern technology proves that it came about between the 1920s and the 1930s. So that's a very long time ago. So we've had HIV on this planet since 1920, and we only became aware of it in 1981. So we also know that it didn't exist before 1910. If it did, it would have come across the Atlantic to the Caribbean and America in the transatlantic slave trade. And we know that didn't happen. So there was no HIV before 1910 because nobody in the slave trade brought HIV to the New World. So the evidence so far is a cross-species infection with SIV is responsible. It adapted and became HIV, and transmission then became epidemic. And the idea is that human beings were interacting with monkeys either for fun or for food and got infected with SIV, and Bob's your uncle became HIV. There's a problem because SIV is not a zoonosis in human beings. In other words, if you catch SIV by accidentally handling a monkey, in the human being, it's a benign infection. So you clear it within days. So SIV is not HIV, and it's not a zoonosis. So we know if you catch Ebola from whatever, a monkey or a bat, Ebola is Ebola. It's Ebola in a monkey, it's Ebola in humans. If you catch Lassa from a rat, it's Lassa in rats and it's Lassa in humans. If you catch Zika from the mosquito, Zika is Zika. So what happened? This is actually a very unusual situation where a human being catches one pathogen, and in the human being, it changes to something else. I mean, it's unprecedented, really. So for SIV to become, I'm sorry, I'm having problems with this. For SIV to become HIV, it needed to acquire several thousand mutations to overcome the human immune system to become HIV. And this is actually mathematically impossible with one single infection. So if I caught SIV from a monkey, it would take thousands of mutations for it to become HIV. So that's not possible. So human technology these days we couldn't edit a virus. So there are various conspiracy theories that it was man-made. In those days, there wasn't that kind of technology. So we couldn't have made HIV. We can make it now. In fact, we can make pathogens worse than HIV, and they do exist under high quarantine, but we couldn't make it then. So that conspiracy theory is dead. So what's the hypothesis? SIV could not possibly naturally evolve to something more virulent in humans like HIV if it was artificially able to evade the immune system. An abnormal environment was required for SIV to acquire thousands of mutations that would enable it to evade a very astute human immune system and overcome that immune system. So that brings us to a little bit of history. So a lot of you know about King Leopold of Belgium. He colonized Congo in the early 1990s, and what was he looking for? He was looking for rubber and timber to drive the Belgian economy. And to do this, he required to have huge concentration camps of manpower. So thousands and thousands of Congolese, millions of Congolese were pushed into these camps in the forests to retrieve these minerals that he was looking for, these resources. And it also permitted a very abnormal environment. And this is a sort of cartogram of the time frame, and you can see from the time the king colonized the Belgium Congo, which is now the DRC, it is estimated that close to 10 million people died in that oppressive regime, and the conditions were just so atrocious that it is hypothesized that that was the breeding ground for the evolution of SIV into HIV. Something else was happening at the time, and it was the advent of the hypodermic needle. So prior to that, we didn't have the capacity to inject. But if you remember, I think I have a slide, we had these kind of needles that were made from glass. And you use the same needle over and over and over and over again. So these were the concentration camps full of Congolese people. There were huge atrocities. They were malnourished. Millions of them died. They were mass vaccinated with these syringes to try and keep them alive. They were catching syphilis because there was a roaring sex trade going on with these thousands of men in concentration camps for their recreation. They would bring a few prostitutes at the weekend. And so there was this constant sexually transmitted diseases, and then they were giving them penicillin and vaccinating them with these injections. So we had a situation where we created that environment, and the immunologists that are here today know what's called serial passaging, where you can take blood from one person and pass it to several people in a very short period of time. And in the process of doing that, you're taking SIV from one person to another person to another person, and that is the perfect environment that is required for the evolution of a virus from one state to another. And that is the current hypothesis for the evolution of HIV. So you can see here in Kinshasa where this was happening in the 1920s was the origin of HIV-M, which is the virus that has populated the world with the pandemic. And then it spread by the trade routes. It went up north, it went south, it went to the coast, and the sailors that were coming from Europe were picking up the viruses. Of course, they were having lots of sexual interactions with the very active prostitution ring that was going on at the time, and they were taking these viruses to Europe and to wherever they were going. So the conditions in the Congo to facilitate this evolution of SIV to HIV in the 30s was the pooled plasma trade. Not many people know about this trade, but it was where blood was taken from a lot of people and the plasma was pooled, and the hemoglobin was given back to the donors, but not in the order in which it was taken. So they just grouped the blood and gave the red cells back, but they were after the plasma because the plasma contained polyclonal antibodies, which was what the blood trade was looking for. Then the hypodermic needle, the vaccination campaigns using single syringes. Unethical animal to human transfusions. There is evidence that some people were experimenting giving chimpanzee blood to human beings to see if they could transmit monkey malaria to human beings. So these were scientists that were working in the region. Dislocation of the family structure, lots of prostitution, severe malnutrition and stress, and several blood transfusions. So that was the environment in which the serial passage of SIV became HIV, and then it became the global pandemic. So HIV evolution was abnormal. It was due to a complete breakdown of human ethical behavior leading to a great biosecurity and ecobiological accident. It's strange that nobody's ever asked, where did HIV come from? It's not in the ecosystem. It didn't exist before, but it's there. And it's caused a global pandemic of, abnormal proportions. So the probable roots of initial global spread in the 60s, this is debatable, but there were lots of events going on in Europe, in America, through the Caribbean. Some people say Haiti. You heard Patrice talk about scientists that came to the Congo and then went back to Haiti. At that time, I believe that the HIV virus itself wasn't that virulent. So it took a little bit more time for it to become the virulent strain that we know it to be. So you all know that the Helsinki Declaration only came into being around 1975. So before 1975, we as scientists could do what we felt like doing. You could do anything you wanted, particularly in Africa, where there were just no rules. It was divided up into colonial territories, and everybody just did what they felt like doing. And that gave rise to this global pandemic of immense proportions. You can see here in the Central Africa, you have every type of HIV, which confirms that that is the location of the origin. And then it spread out to affect global populations based on demographic and social pattern behaviors. So they say it costs about $1,500 to manage a patient a year in South Africa. That's cheap. Now, 10, 15 years ago, it was a lot more than that. But let's just work on this figure. $1,500. What is the value of life? I've spent a bit of time trying to research this. And I've arrived at $3 million. Now you could say, how could you even tell me that life is worth $3 million? Well, for some people it's worth a lot more, depending on your social contribution, to society, your value, your ability to create. For some people it's less, like King Leopold has a deficit because he caused so much pain and suffering. But the average person, I imagine, has a value of 3 million. So if we look at the total cost to Africa of HIV, 1,500 for 10 years at 30 million people infected. The cost of managing people in Africa with HIV is 45 billion per year, just managing. If you look at the people that have died, 30 million have died, roughly. If the cost of each life is 3 million, it means Africa has lost $90 trillion. I Googled the Global Fund just to see what the world is contributing to the eradication of HIV. And I was shocked. The biggest contributor is the United States at 1.5 billion for the Global Fund and 7 billion for PEPFAR. Thank you, America. We really appreciate it. But Belgium, 16 million. When the target is 45 billion for care and 90 trillion for deaths. So we shouldn't even be talking about aid. We should be talking about compensation. And this is the value in my simple mathematics. I'm not a mathematician. I'm sure it's probably worse than that because you can't compensate for pain and suffering. So how can Belgium, that contributed to build its economy and cause HIV, be given 16 million per year to the Global Fund? It's just absurd. And I think we need to look at it from this dimension. We shouldn't, in Africa or anywhere that is inundated with HIV, be talking about aid. It's a time for reparation. It's a time for compensation. So what has this got to do with cure? Well, if Belgium would pay for the damage and compensate the world, and Africa in particular, to the tune of 90 trillion for contributing to the death of 30 million people and growing, then people like George and Frank, Mandy, Clive, and some of the brilliant minds in Africa will go about finding a cure. And I assure you, with that amount of money, we'll find a cure in 6 months and put an end to this epidemic. So, in 2012, the Society for AIDS declared that we were going to embark on an HIV cure strategy. So, these are some of the concepts. Understanding the genetics of latency in how humans handle the virus differently. Mimicking the SIV latency in monkeys. Early and prolonged therapy. The stem cell transplant with the Delta 32, I'll explain that. You watched the video of it earlier on. I'll try and explain that. The latency can be eliminated therapeutically and then genetically engineered stem cells. So let's dream a little bit now. So the pathogenesis of HIV, I'm sure a lot of you know this. It's possibly, there's some clues there. Many people talk about CD4. And if you haven't seen this slide before, we follow this CD4 count in the blood. It's called the circulating CD4 count. But that's just a very small proportion of your CD4 count. Your main CD4 count is in your tissue. It's in the mucosal surfaces. And look at that. In the first month of acquiring HIV, your CD4 repertoire is wiped out completely. This is the real problem. It's just like someone who's gone through university and you have an accident and you lose your memory completely. You don't even know who you are. You don't know your profession anymore. You don't know anybody. You have to go back to kindergarten. This is what's happening in HIV. We're losing our immunological memory completely. And then we have to rebuild it once we start hard. Forget about this blue line. That's not the reality. This is what's really going on. So if we can start treatment at the point at which you actually acquire HIV, then you can stop this process. And how early is early? I can't remember who presented the slides I think, you don't catch malaria and wait a couple of weeks before you treat it. You catch malaria and you treat it immediately. So if we can identify what the symptoms are of acute HIV and make people aware that you've had a risky encounter, you now have the symptoms of HIV, go and get treatment within 24, 48 hours. Perhaps we can address this assault on the immunological repertoire of the body. Immune activation and exhaustion. This is Clive's pet subject. We know that every patient has a different profile. If the natural history, we've had so many millions of people untreated. So we know the natural history of HIV. It's not unethical now to talk about the syphilis trial where they try to understand the natural history, the unethical trial. Here we know the natural history because of the years of inaccess to heart. So we know that some people have a different gradient in terms of CD4 loss. So why do some people lose their CD4 gradually and some have a very precipitous fall? So now we're in the age of genomics. We can divide all the HIV patients. If all these people have HIV, we can divide them into different categories and say these are perhaps the slow progressors, the medium progressors, the fast, the rapid, and the accelerated. We have access to these samples now. So we can put that through genomic whole exome sequencing, apply some bioinformatics, and then try to understand what are the drivers of slow progression as opposed to rapid progression, and then come up with some kind of small molecules that interfere with the process and convert all rapid progressors to slow progressors. We also now think that we can try and mimic the natural resistance to HIV using various technologies, including bone marrow transplant. So we know that there are chemokine receptor families, CCRX, whatever, and in the monkey, if we can change, this is the monkey status, it's living with the virus as opposed to dying with the virus. In man, we're living in a hyperactive state in HIV, which is what kills us. But the monkey is very happy living with its SIV virus. So how is the monkey able to be in this state? So we as human beings with HIV need to find a way from going to the hyperactive state to the underactive state of the SIV model. So that is a whole lecture in itself, but suffice to say that there are some ideas coming up in that direction. Now I want to focus on the CCR5, the chemokine receptor 5. It's present on the cells of human beings, and there is a deletion called delta 32. And if you're homozygous for this natural inherited mutation, then you are almost resistant to HIV. You can be exposed to HIV as many times as you want, you just won't catch it. So the reason for that is because the HIV requires two receptors. It requires the CD4 and the CCR5. Now, if you're missing CCR5 as a genetic part of your heritage, then it doesn't matter how many times you're exposed to the virus, you can't catch it because you need both. So this cartoon shows that if you have CCR5 and CD4, the virus can infect your cell. If you're missing the CCR5, what's called the delta 32 homozygous state, then you're deemed resistant to HIV. So what would the possible indications be for us to contemplate doing a transplant using a delta 32 donor? So for example, we could think about what is the economics of lifetime treatment of heart versus one procedure that takes a couple of weeks. It might cost us $20,000 to do the procedure, and then you're done. You're cured, as opposed to 70 years of taking heart. So, the health economists need to look at that. Patients with dual pathology that could benefit from a transplant. So we have a lot of lymphomas in HIV, and we treat lymphomas with transplants. So we could, kill two birds with one stone, cure the HIV and cure the lymphoma. Patients with severe heart resistance, and patients who will just not adhere to treatment, and you want to take them out of the pool so that they don't become a source of continuous infection. So this gene, delta 32, gives you the profile of being resistant to HIV. And it is believed that the highest density of this gene is present in Europe. And they feel that it gave the Northern Europeans protection against the bubonic plague or smallpox. And that's why it is so prevalent in Europe and is almost absent in Africa. Hence, there is no genetic inherited trait related to CCR5 that protects the African community. So if you have this mutation, it's 32, it's called delta 32 because it's a shortened version of the wild type. And when you have delta 32, your CCR5 receptor is on the surface, it's shortened, and there it doesn't express itself on the surface, or at least the density is much lower. And so the virus can't dock. And there you have it. You have a situation where the wild type, the receptor is on the surface, and the genetic delta 32 is inside the cell, so the virus in someone who has delta 32 cannot be infected by HIV. So here you could be homozygous or you could be heterozygote. If you're homozygous, you're highly resistant to HIV. If you're heterozygote, you will catch HIV, or you could catch HIV, but you would be a slow progressor. So that's the situation. Now, you've heard about the Berlin patient. So this was a patient who had HIV, but also developed acute leukemia. He was 40 years old. He was on heart. He developed acute leukemia. He failed standard leukemia treatment, and his only option for survival was to have a bone marrow transplant. So they found donors, and it so happened that one of his donors was a delta 32. So his doctors were very astute in Germany, and they said, wow, let's pick the donor with the delta 32 and let's see what's going to happen. So this was a clinical experiment. And so he had his transplant. So you get the donor gives you the bone marrow, and then you give it, so that's the bone marrow, and you give it to the patient. He had his transplant, and that's just a concept explaining how a transplant works. And eventually, the recipient cells are overtaken by the donor cells, and after a period of time, the recipient has the cells from the donor and none of his own original cells. So that's the principle. So we took blood from the donor who was delta 32. We gave it to this patient who was acute leukemia with HIV. He recovered from his leukemia, and that's just a cartoon showing how it happened. And over a couple of months, his viral load went to undetectable, and his CD4 count went up, and he didn't need to use heart anymore. And just to prove that his tissue CD4 count had recovered, they did a gut biopsy, and they found that he had repopulated his gut with the donor lymphocytes. So his leaky gut syndrome had been repaired by the transplant, which is a problem, which is the cause of HIV in terms of its pathogenesis. So the conclusion and significance, the role of CCR5 co-receptor is vital for maintaining HIV infections and disease progression, and based on overwhelming positive results of this case study, we now know that CCR5 and various drugs have been developed, and you've heard about them, But the interesting thing is that by performing this transplant, we wiped out his reservoir as well. So if you don't have CCR5, you actually eliminate your reservoir. So I'm collaborating with a foundation called Cure AIDS, which is based in America, and we're trying to use in Africa this concept whereby we're going to try and manipulate cells to remove the CCR5 receptor and use that to replenish the HIV patients and see if we can cure people. And we even want to go a step further and create a stockpile of various immunologically profiled cells so that people can come to a facility, have a procedure for about a week or two as an outpatient, and we can convert them from HIV positive to HIV negative with elimination of their reservoirs. So there are major ethical issues involved here. I mean, you don't just start a program like this, and in my university, we've started a whole ethical campaign around cure for HIV, not cure for AIDS. We've heard that these are two different things. Because the moment you start talking about cure for HIV, there's going to be a public arousal. And you've got to know what you're talking about. You don't start talking about something for which you can't deliver. So we've started working on public campaigns to just educate people about the possibility that we have the technology now to find a cure for HIV, but we just don't have the money. We don't have the infrastructure. So we could mimic this by genetically modifying cells, and the technology is advancing so fast. We had zinc fingers, now we have CRISPases, and we have oligonucleotide technology, and cell sorting is just advancing so fast that we are on the brink of being able to perform this kind of cure using this kind of genetic editing of cells. But we have to be careful because knockdown CCR5 is not, there's a reason why CCR5 is not completely knocked down. So maybe CCR5 is important for some immunological mechanisms. So it would be important that we try and mimic what's happening in life rather than knocking down the gene completely. So the idea is that we try and recreate the natural type of delta 32 in these stem cells and then reintroduce these stem cells into patients. So we would take blood from a patient, we would manipulate them in the laboratory, and then we would give them back so they would now be CCR5 negative, but mimicked to what's actually really happening in life, not just knocking down the gene. And then hopefully that would result in the patient becoming HIV negative, just like the Berlin patient. So there are other technologies now. You've all heard about the iPSC technology. You can take skin from a patient and you can convert those cells into embryonic stem cells. You can manipulate those stem cells, knock down the CCR5 or mimic the CCR5 and then give them back to the patient. And this technology is very, very promising in terms of its therapeutic modalities. So you might ask, well, in Africa, where there are 40 million people living with HIV, is there the infrastructure for bone marrow transplant? Well, there is. It's not everywhere. This is the unit that I'm involved in. And this is the apheresis unit where we harvest the bone marrow stem cells. And there is a patient whose stem cells have been harvested. And this is our transplant unit. This is one of my students there, and the nurses. And this chap is doing quality assurance. And so there is the capacity. There are about 5 such units in Africa. But we're not talking about transplant for oncology. We're talking about transplant for HIV therapy. So it's not a myeloablative process. And so this could possibly be done on an outpatient basis. So the technology is there, the infrastructure is potentially there. We can create more of these. If we had the money to embark on this research, this could be reality. Now, we heard about sanctuary sites. So when you've had your treatment and you're undetectable in terms of picking up viral load in your blood, but the virus is still living in sanctuary sites. So how do we try and find these sanctuary sites and monitor them if we're going to develop a strategy to kill the latent viruses in these sanctuary sites? So we use one technique called sushi, fishing for reservoirs, using fish and flow. And how does it work? So you know when the virus infects the cell, it's an RNA virus. It converts its RNA to DNA, and the DNA incorporates itself in the human genome permanently. So what we need to do is we need to look for these inserted HIV DNA in the cells, and you can do that with a combination of flow cytometry and probes, and then you can determine which cells are negative and which cells are positive if the probe detects the DNA of the HIV in the sanctuary cells. So we can use this technology with some very clever flow cytometry and try and figure out which cells are the reservoirs of HIV, so the memory cells or the effector memory cells or the whatever compartment these viruses or the monocytes. So we can find out where the reservoirs are, and we can do some elegant research to try and figure out where the reservoirs are. So this is some work that I've done with the lymphomas, and just to point out that we had a rising incidence of lymphomas in South Africa, almost doubling every year, but that has tapered off and is now decreasing because of the national policies for treatment. So here it was treat at 200, then it was treat at 350, then it's now treat at 500, and soon is treat on diagnosis. So you can see how policy is influencing some of the oncologies that we see in HIV. This is some of my research team in Cape Town. This is a bioinformatician, this is a programmer, PhD student, a molecular biologist, a pathologist, and a cell culture expert that work on some of these projects. Now, you thought that the picture of the lion and myself was fake. Well, that's a real cheetah. We're looking for FIV virus. And some of you think that the lion is the most dangerous animal in Africa. It's actually not. It's one of the softest animals in Africa. The most dangerous animal is the buffalo. This animal is responsible for killing more people than any other animal because it's a very aggressive animal. And here we're looking for bovine TB as we move buffalo from one region to another. So this, quickly, some of you have been asking what I'm doing since I left Barbados. Well, I've been involved with the Ebola outbreak, and this is just a cartoon of how the Ebola outbreak spread across 3 countries over a very short period of time. And you can see the epicenters. This is Guinea, Sierra Leone, and Liberia. And within 4 months, it had escalated to 3 countries. Never done that before, so this was an eye-opener for us. And it caused the collapse of the economy of these 3 countries and of the sub-region and of Africa as a whole. So emerging infectious diseases are not trivial. The Ebola outbreak infected at least 28,000 people, killed 11,000, and there were 14,000 survivors. And so we formed this organization similar to CCAS, but this is the equivalent of CCAS in Africa. It's called GET, Global Emerging Pathogens Treatment Consortium, and these were its objectives. And it quickly expanded with experts from across the continent, and now we have almost 200 members. and we got engaged in a lot of community activity. Here are all survivors from Ebola. That's me up there, and these are all Ebola survivors. And what were we doing with them? We were engaging them because we wanted this. And this is white gold. It's plasma. And why did we want plasma from survivors? Because it contained the antibodies that help them to survive Ebola. And there is no treatment for Ebola. So this outbreak was expanding out of control. We didn't know what to do. It was threatening to consume the whole West African region. So we set up a huge organization that had these, we parked these mobile units outside these treatment units. So this is an Ebola treatment unit where thousands of people were in there. Many, many people died in this treatment unit, but some survived. And so we would harvest the plasma inside this facility. And these are some of my survivors that were working with me. If you had a t-shirt called "I Made It," so she made it. These are all doctors. He made it. And these are all volunteers. And we were able to harvest plasma from these people and use it as the only means of treating Ebola. At the time, we used both plasma and blood. This is my colleague, Professor Javal, who worked with me on that project. I'm showing this slide because I'm eternally grateful to Clive. When we started CCAS here in 2004, or was it 2006, I'd been here already for about 10 years, and we knew the importance of CCAS, and you've heard the impact that CCAS has had across the region. But I had to go back to Africa. And so I asked Clive if he would take it over. And not only did he take it over, but he's taken it to great heights. And I just want you to give Clive a big round of applause. Clive is a really nice guy, but I can tell you he's a serious dude. And don't mess with Clive. So we have our conferences in Africa as well, and I'm hoping that some of you will come and join us. We just had the third African conference on emerging infectious diseases and biosecurity in Accra, Ghana. We just finished 2 weeks ago. We're going to have the fourth one, probably in Sierra Leone, but I will send all of you information on this. I like this saying by Rumi, you start to walk on the way. As you start to walk on the way, then the way appears. I think it's very apt. I know that I coined this in one of my talks, and Clive really liked it. Another pseudonym for AIDS, advancement in infrastructure, diagnostics in the third world, and softening the hearts of capitalists. These were, they call them Akin's angels, and Dr. Jerome Walcott is sitting here, the former Minister of Health. Thank you very much for coming to this. Without Jerome, LRU wouldn't have happened in the way it happened, and Lady Mead is where we all were able to get together. This is a very youngish me, and surrounded by Akin's angels, Nami, Kelly, and Sonji, and Clive joined us, and these were really the starting point before Andrea and Co. and Vera came, and those of you that knew CCAS meetings earlier on will remember Nami just had a baby, so I imagine that's why she's not here. And this was the kind of work we were doing in the laboratory. We were introducing flow cytometry, trying to use it. This is the flow cytometer we had at the back there. That's Shona, right? And what's this girl's name from Trinidad? Huh? Who?
Arlene.
Darlene, yes. So on a serious note, this is one of my last slides. We recognized after Ebola that most countries do not have a biosecurity agenda. And that is why we're being consumed by these emerging infectious diseases. And HIV was caused by a complete absence of a biosecurity agenda in the early 1990s. But now Ebola has opened our eyes, and it is very important that each country develops a biosecurity agenda. And there are various parameters to a biosecurity agenda. I'm not going to go through it, but it requires some thought. It requires some adaptation to your local environment, and it's made up of policies, guidelines, processes, and adherence to various international treaties like the Biological Weapons Convention, the International Health Regulations, the Global Health Security Agenda. And it is imperative that the Caribbean develops its biosecurity agenda. And each country needs its own, because every country is different, but they can be adapted. So I'm glad that Jerome is here. I'm glad that people from CAFRA and various countries across the Caribbean are here to listen to this. We need biosecurity agendas. Otherwise, we're going to run into problems. And I thank you for your attention.
Thank you very much for that compelling lecture. The floor is open for discussion, questions, comments, etc. I think you've challenged us all. George, please use the microphone.
Quite an astonishing series of talks which contain 3 different parts, which all would need separate discussion for hours. The first feeling to me was a flippant command which I would have kind of imagined to document my sense of humor, because I was thinking that you were producing that plan for the bone marrow transplantation just because you are a hematologist and you wanted to recruit more patients into your department. But then the smile has kind of vanished from my face, because the details which followed in terms of the individual steps and the widespread relevance of the suggestion has suddenly showed that actually it contained a number of feasible looking and very important research projects which cannot be flippantly dismissed. And although I think we would all agree that this is a very high kind of request for any communities to consider it, it's still an incredibly important revolutionary suggestion. The amazing thing in it is that you have the capability of actually executing quite a lot of this work in your present situation. And so therefore I was extremely impressed by that. Now the first part was frightening, because putting the blame straight away to the Belgian Leopold, poor sod, he caused really a lot of trouble, and the Belgians should be ashamed of themselves. It's actually bringing up such an atmospheric and huge problems which are relevant in the current situation. I'm obviously living in England and in Europe, and the current developments in the American internal politics is also bringing up these kind of issues of handling people in a mass scale. The current strives in Berkeley and other places with the taking of the status of those who have been slave keeping diplomats and that sort of thing brings up the situation is that if the USA would consider paying compensation for the black people who were transferred from Africa just because on the south it was impossible to grow without having workforce who were so incredibly well constructed that they were the only people who could stand these conditions. And if the blacks would put in demands for the government for compensation, it would break the American budget. And it's exactly the same for the Europeans of handling with Belga Congo and that kind of thing. So if that is related to the unbelievable expansion of military spending, of course, Steve Bannon was trying to kind of persuade Trump to have some kind of suggestion that the military involvement should be stopped, then this thing is all of the window now. So that kind of thing of paying Saudi Arabia this kind of 57 billions when the American budget is multi-trillion, be in a negative, in that kind of situation, we should certainly realize that there is something wrong. And that means that when we are considering the AIDS-related budgets, when that sort of expenses and misbehaviors are happening, we should never be ashamed of asking for more support and more money when there are backed up with such an incredible efforts which have been documented here. So therefore, the blame which we put on previous governments should encourage the HIV fighters here to be seriously supportive of further expansion of the HIV-related work. And I think this was an extraordinary span which you documented, and I just kind of trembling that how much you can be able to bring through, because the quality was so phenomenal that it certainly must be rubbing up quite a lot of the souls of your South African leadership.
Thank you, George. Clive?
Thanks, Akin. Just a couple of questions. I was very interested in your experience with Ebola. Do you have results from the use of the plasma?
Yes, so the logistics for putting in these mobile units into the heart of the outbreak, which was called the red zone. And remember that all three countries were shut down. There was no flight in, no flight out. You could only get in with the United Nations aircraft.
Yeah.
So we had to fly in these equipment, train, do the community engagement, get the survivors, and I could talk for 3 hours on that topic. But by the time we got the plasma, the outbreak was already receding. But we had started a whole blood project, and the whole blood project demonstrated effect. And there's no reason why the plasma shouldn't work.
Okay. One other question. I wasn't quite sure what you were saying about the editing of these stem cells. Were you speaking about a deletion editing, or were you speaking about a knockdown of the gene? I wasn't quite sure what you were advocating.
Right. So I think Sangamo are working on deletion of the gene, but we're proposing that we mimic Delta 32, which is not a gene knockdown. It's a shortening of the gene so that it remains in the cytoplasm, the protein, the receptor remains in the cytoplasm, but can't dock with the membrane. And therefore, the concentration of CCR5 on the surface membrane is drastically reduced, and that seems to be enough to stop the virus from spreading from cell to cell and to eliminate the reservoirs. So we're talking not about knockdown, we're talking about mimicry here. And there are many technologies that are evolving. It's mimicry plus cell sorting. The flow cytometrists in this room know that it's just as difficult to cell sort.
And also just thank you for concluding the expert summit, I think you all now know why I pick Akin always to be the last speaker.
Akin, thank you for a wonderful talk, as always. In another life I was into transplants and so on. So I actually wanted to ask a question about what happens in HIV with respect to the histocompatibility antigens, because you talked about the fact that graft versus host disease is not a problem. So I'm very interested to find out exactly why that is when you're looking at transplant, into HIV positive persons.
So graft versus host is actually what we use. It's the tool of transplant. It's not the actual transplant that does the curing. It's the graft versus host. So the graft versus host is the incoming immune cells set up an immunological reaction to the host because there's a little bit of mismatch. So the donor cells recognize the host as foreign. So that sets up graft versus host. Now, if you have a leukemia or lymphoma on board, that graft versus host is what kills the leukemia and what kills the lymphoma. Now, we think that the graft versus host also will eliminate the viral reservoirs of HIV. So we're thinking that graft versus reservoir is a phenomenon that we're dealing with here, or in part.
But you still have to match.
Yes, you still have to match. When we talked about creating banks, we talked about creating, just like you have a blood bank with different groups, you can create a nice repertoire of thousands of HLA types. And your patients would just come in, you would match them to a reservoir that you have, you've engineered them, you've got them in your biobank, they come in, you give them a gentle myeloablation, which just creates a little bit of space in the bone marrow. You give them the incoming manipulated graft, which can be theirs or can be somebody else's. And then that populates the bone marrow, and it has an advantage over the other cells because they can't be infected with HIV.
Thank you.
I'm just going to add a comment in the interest of certain inside information a little bit. The Sangamo trial, sponsored by City of Hope in Los Angeles, is currently recruiting participants with CD34 zinc finger modified CCR5 receptor. So that is active and opening as of June of this year.
Is it knocked down or modified?
It's modified.
Okay.
And so it's not eliminating, as you said. It's the modified zinc finger.
And I think I read some of the phase 1 results, and they were a little bit promising. At least toxicity wasn't an issue.
Exactly. This is still a feasibility pilot for safety and toxicity. So it's still a phase 1, even though it's their third round of their zinc finger nuclease attempt to knock down CCR5.
Okay.
So we can look forward to the results of that clinical trial.
Yeah, thanks for an amazing presentation and an education in itself. Just a question. Selective pressures are almost impossible to predict. You never know when a particular gene might be an advantage or a disadvantage. Once we start manipulating a particular gene to protect against a particular pathogen, we also create the opportunity for another pathogen that will use the opportunity of that manipulation to cause problems. Is it the knockdown approach? It keeps the functionality of the chemokine receptor sufficiently so that there's less of a risk of that? Or what about removing it long enough to remove the reservoir and then giving it back?
Yes, I can see you're a true immunologist. So there's always a give and take. So just like in sickle cell disease, it's prevalent in malarious regions because the carrier confers resistance against malaria. But then the downside of that is that you have thousands of millions of kids with sickle cell homozygous disease. So there's always a trade-off. So if you knock down CCR5 in everyone else is present. So it must be there for a reason. So what is CCR5 doing, and what would knock down CCR5 predispose you to in the long run? So there is evidence that if you knock down CCR5, you predispose yourself to certain types of encephalitic viruses. So there might be a price to pay, but the people who have CCR5 truncated don't seem to have a problem, which is why we're opting for truncation rather than knockdown. So if you truncate CCR5, it remains present in the cytoplasm. Its density on the membrane is much reduced, but it still seems to confer whatever physiological function it was designed to do in nature. So we're opting for truncation rather than knockdown, thereby minimizing the long-term consequences of genetically modified human beings. So we're talking about here, we're talking about gene editing, which is modifying. It's not alien anymore. Many people are going through gene editing as a therapeutic modality. So, Chairman?
Just one other concern for the record. When we do the gene editing, my understanding it's not as clean as just snipping out a gene. We do things that could disrupt neighboring genes, and we have no idea what's happening in those zones that we disrupt. So we can also be activating, opening all kinds of Pandora's boxes in the process. So I'm just walking a cautious line with respect to that approach.
Yeah, so gene editing is called collateral effect. So you can't always target the zinc fingers to exactly the point you want. So sometimes you get collateral activity which goes and switches on or switches off other genes. But the technology is advancing so fast and is getting so specific that you can actually pinpoint exactly where you want to go and where you want to edit now. And the oligonucleotide technology is also very elegant, which doesn't cause collateral impact. So technology is really advancing. So very soon, we're going to be able to gene edit with precision.
Can I comment on that?
Wonderful talk, Tom Quinn from NIH. I really enjoyed it. The first question I have is on the Ebola and getting the plasma from the survivors. There's a big survivor cohort study now underway looking at the downstream effects Ebola's had on these individuals, and some of them have had Ebola present in retina, eye fluids, and semen and other areas. Is that, I don't know if you're continuing to follow the survivors and what you might have found. Some have had neurologic effects. We're really just learning the whole paradigm of Ebola and what a survivor has to go through if they make it through the original illness.
So we were not aware of this post-Ebola syndrome before because the outbreaks were so small, and just the outbreak died away. A few people survived, and they were irrelevant because they lived in the jungles of Africa, and who cared? But now, in Ebola that cut across 5 countries, affected thousands of people, we have 14,000 survivors. And now we recognize that there's a syndrome called post-Ebola syndrome, which is a spectrum of neurological, psychological, all kinds of phenomena that are going on. So the post-Ebola cohort is active in all 3 countries, and we're beginning to try and understand what are long-term sequelae of Ebola. But I guess your question is, in harvesting the plasma, is there a risk of harvesting active Ebola virus? And if we use that, well, we wouldn't be using it in any other person except that person had Ebola, 1. And 2, we put it through what we call a viral reduction process, which there are many technologies now. The one we used was the Sorolin UV light. So you put Sorolin into the plasma, It intercalates with any nucleic acid. So if there's a virus or bacteria in there from any virus, known or unknown, and you shine UV light on it, then the psoralen absorbs the UV light and destroys the nucleic acid. So that way we can render the plasma completely free of any pathogen, known or unknown, as long as it has nucleic acids. I'm not talking about prions now. Nucleic acids will be killed by the viral reduction process.
So that would render it fairly safe.
Yeah.
One other area that is pretty recent, just published in Science about 6 months ago, is looking at the integrin, alpha-4 beta-7, which is another receptor for the virus. And they did experiments in primates and used a monoclonal antibody that binds to the integrin. And while on that, they could stop the antiretroviral drugs in the primates in the study that was being done, and there was no rebound. The reservoir is still there,
Yeah.
but for some reason there's this interaction. So I'm not quite sure where it's going to go, except that it's in phase 1 trials in humans right now at the clinical center, and we'll see what the safety parameters are. But as I was listening to your talk about the editing of CCR5, whether that works or not, I think even downstream there may be other opportunities for other receptors of the virus that we might be able to look at.
Yeah, sure. And in gene editing, sometimes you don't edit one target, you edit 2 or 3, because viruses are adaptive. So for example, if you edit CCR5, the virus could change to a CCRX4 and just bypass, it will evolve with your technology. So you need to, just like combination therapy, you might need combination gene editing to have a really fail-safe technique. So it could be a CCR5, a CXCR4, and alpha-4 beta-7 target.
Right, thank you. I mean, as another editorial note, gene editing is exploding. I mean, you referred to it a few times. It's growing like crazy. The publications are growing exponentially. The applications, the technology. For those of you that are interested and want to kind of get involved, there's a book out by the discoverer of the latest CRISPR-Cas9. It's called The Crack in Creation, Gene Editing, and the Unthinkable Power to Control the Human Evolution. And it's fascinating. It's this woman scientist and her PhD student who have written the book and is hypothesizing on how far you can go. I mean, everything we're talking is not germ cell. It's all somatic cell editing. But if you can imagine editing the germ cell, you could potentially change human genetics. And so it's a fascinating read. It gives you a little introduction on how all of the gene editing technology evolved, et cetera. So that's just a little editorial aside. It's a fascinating and easy book to read, and you can read it in a couple days. So I want to thank the speakers for this afternoon. I have one housekeeping comment. If you could all please complete your evaluations, we would be most grateful. And with that, I want to thank Akin and Eric for fabulous presentations to end the day's session.